Finally, it's just for you a colorblind test. So if everyone can distinguish between green and orange, then you are not colorblind. So I will not look in details at the history and the evolutions of extractables and leachables, just to mention, and thank you, Mr. Jenke. I can tell you, your child, 665, is really welcome and is helping to clarify the horizon about extractables and leachables, definitely. Here, it becomes something that is really clear, that is a really regulatory expectation and not something that people need to pick up and hope it will be accepted by authorities. And just to mention, I don't know if it's a magic 85% number, but a recent survey from Bioforum concluded that 85% of the respondents considered that USP 665 is providing the adequate response to regulatory expectations about extractables and leachables. So, great. So just to say also, as Simon mentioned, we have soon 665 that will become effective in 2026, followed basically a year and a half after by 383. So we have a lot of stuff to be able to type numbers now. But also that it's not because it's effective in 2026 that is not currently an expectation. Both we heard a lot of authorities, and not only FDA, are looking at and taking 665 as reference, but also that, of course, for a lot of end users, this need is there now because they need to prepare themselves to make some gap assessments like was brilliantly shown by GSK, or to prepare new filings where they will have to be ready in tomorrow, basically, in 2 years' time. Just brief overview, and I will not speak specifically, I think, about Cytiva later, just to say that Cytiva has a global footprint, and that, as we heard, we need, and there is a need for to be compliant with USP 665 to leverage supplier data. So basically, we have a lab on the 3 continents where we are able to perform and where we are performing extractable profile testing, but also labs where we are performing less specific testing. And you see here a small image of that. And of course, on top, and we heard that if 665 is really too worst case for you, you can still use simulation studies or leachable studies to generate the data that is needed to build an efficient and relevant risk assessment, tox risk assessment, sorry. So what is the approach of Cytiva? And I don't think we are reinventing the wheel here. It's quite consistent with what we heard in the previous presentations about basically we need to consider first the single-use system, but also what is not dissociable from the single-use system, its intended use or its application, of course. Then basically based on the application, you can build a risk assessment. We heard about BPOG best practices for risk assessment that was published in 2017, or the 1665 now that is more recent, and to which we currently in Cytiva align with because it's more recent and also more associated to regulatory expectation. That doesn't mean that the BPOG is not acceptable, definitely, like Simon was mentioning. It's just that we feel that more and more customers will look to the source and not to something that is maybe as relevant but has not the badge USP. Based on that, we derive a risk level, and the risk level will define, as we have seen, the dataset info level, the level of extractable information you need. And like a lot of suppliers, we are working on a component-based level, because when you have a few thousands of components in your portfolio, if you have to test each and every single-use system you can generate, then even with a very cheap analytical method, you can very easily, quickly get bankrupt. And anyway, the time needed will not be enough till the next millennium. So, with that, with the component dataset and the info level you need, basically you derive exactly and you extract from your database the extractables dataset. You aggregate it and scale it according to your exact single-use system, and then you are ready to perform your tox assessment. If everything, and in the nice world, everything is okay and it's done. It's not a tick of the box exercise, by the way. I don't want to mean that, but at least it's okay. Now, sometimes you still have question marks, even with the best defined, performed study. Sometimes you still have unknowns, for example. I remember some years ago that an unknown killed, let's say, an active ingredient storage container when I worked in GSK, just because this unknown was at a level that was not unbearable. So it's really important. Also, sometimes you miss some information. I will come later to that. But you can do other stuff like extractables or leachable study if needed. So you don't need to kill everything immediately. About the dataset info level, and that's basically the approach that we are following. And there it's a little mix about the needs associated to a tox assessment and the needs associated to the business. So basically, we are generating, depending on typically where you are in the process, and don't tell me that it's 100% the case here. We also use our brain, and sometimes we adapt the risk assessment. But basically, in early steps and in downstream processing, we consider we are between low risk and moderate risk, and more we are arriving downstream the process, we reach USP high risk, and because of the let's say status of BPOC as industry standards in the past, and because it provides sometimes additional relevant information, really when we are close to the end of the process, we tend to generate both USP 665 high risk and BPOG data. The same for the same way, and targeting some compound, like I heard about some phosphate. Basically, we are looking at the initial steps, cell culture steps, where we generate extensive supportive data. Also, there it's not really for a full tox assessment, it's more associated to the business needs. Risk level, as I mentioned, this is a worksheet that used to be accessible directly on the PAL website. Now that we became Cytiva, we are managing some transition in our internet tools, and this is unfortunately not available directly for our customer. Directly on the website, but this is how we can perform a systematic risk assessment according 665. So now Extractables dataset. Again, you see two different templates on my screen. It's because we are in a process of merging our approach between our two historical companies, Cytiva and PAL. They are anyway both available for our customers free of charge directly from our website. And you can even, I do a free advertising for Bioforum. If you pass through the Bioforum Extractables portal, with a single click, you arrive on the Cytiva Extractables portal that is providing all the information we have on extractables. This is including all of our products, and when we have the permission, you even have access to information from some of our suppliers when they allow us to share that information directly via our portal. Now, what the extractable dataset we are generating, as I mentioned, basically, okay, our standard level is starting at moderate risk, because very rarely we stick to the low risk level. And then depending on where we go, we extend to a high risk and high risk plus bioforum data. We don't necessarily do all the bioforum time points with our new studies. It depends on the case. But we are generating the data for the 4 solvents. And of course, that's to satisfy Mr. Hawke. For some application, you need to go beyond 50% ethanol. And we are, depending on the application, we are also generating some 100% organic extractions. Typically, if you look at mRNA, some steps of mRNA processes or things like that. So the baseline for us, for all of our new product, is at least to generate the 665 high risk. And as I mentioned, where we typically are at the level of fill and finish activities, we tend to generate USP 665 plus the full BPOG dataset. Now, as I mentioned, we don't, of course, generate all the data sets for the components that are coming from our suppliers. Would be nice, and I think GSK would be very happy of that. I see them, but of course it's not manageable. So you need to leverage, and I think everybody heard from the previous presentation that is the case. You need to be able to generate and to leverage, data from the supplier base. And we are necessarily not totally there yet, even if, of course, for big suppliers, namely people like Millipore or Sartorius that presented 3M, they have the power to do extensive and very robust extractable studies. For smaller players, this is a big challenge. And also, sometimes it's even a big challenge to get access to very basic fundamental information, like the surface exposed to the product. Just to give here, in my company, we segment component in, let's say, 3 categories: preferred, non-preferred, and restricted component. restricted as are the ones from our competitors. Surprise. So we can put them on our systems, but of course it's not our preferred choice, but also sometimes that means that we don't have necessarily the same range of evaluation or auditing for these components. Sartorius will not accept Cytiva auditing Sartorius, which is fully understandable, but that means that some information is missing for that stuff. That doesn't mean that we don't put Sartorius product, and Sartorius is selling us product to put on systems when our customers request that. But if you look at the two other categories that are basically the one, or let's say, our standard, I heard sometimes standardization, sustainability, but also standardization in the last presentations. So basically we have the components we push more as standard, and you see that still 31% of them we don't even get the surface area exposed to the product from the supplier of this component. The information, for me, there is no secret. There is no reason to hide it. So we are in a process, certainly, and contacted each supplier to get this information, but it's not something that is readily available. So I don't speak about the non-preferred, that's basically the same. Now, when you want to aggregate data sets, you need to be very careful, and I was happy to hear that even if it may be a tick of the box exercise currently, don't expect that to stay that way. It's the same for everything. Authorities expect that when you leverage data, you do a proper assessment of the quality of the data. And thank you, Alicia, to confirm that that's what you do as end user, and that's really very, very important. And there are some challenges, and sometimes it's very difficult. First, because sometimes this data has been generated before 665 or during the different evolutions of 665, so you don't necessarily have really something that totally answers current 665 needs. And sometimes, basically, there is even no data at all. So that's a first point. Second point, and it comes with very demanding applications. Sometimes the reporting limit of the extractable study doesn't meet the analytical evaluation threshold. That means that you should have a better resolution if you want to robustly address the question of the tox risk assessment. So again, that's something that needs to be carefully evaluated and reviewed. Also, and I have a nice example, one of my colleagues forwarded to me, and I was quite shocked because this is typically the kind of data set I would hate, or I would have hated when I was end user, where you see, and I voluntarily did not put all the names of the compound, but if you look, I hope some of you can see, but all the compounds are either tentative or unknown. Really, good luck to make your tox assessment with that. So really, you need to be very careful when you leverage and you aggregate data. That means also that sometimes it's not because you have too high levels of a compound that you need to run a simulation study. It's just because you don't have the information, and having to generate extractable information for each and every component where you don't have the level, either the information itself or at least the information at a quality level that is satisfactory, then you need to switch to some other solution, more practical, like simulation studies or leachable studies when it's feasible. With a biological product, simulation studies is often more easily done than a real leachable. to avoid the noise of the product itself. So just a small example about, you have on the left a typical single-use system that is final filtration step, and basically on the right the simplified system that was used to perform the simulation study. Basically what was removed was the waste bag that is not playing a role in the simulation, in the extractables. Now if you compare what you will generate with simulation study and what you have when you perform a paper exercise, you see that you will have different sterilization conditions, meaning namely different irradiation dose. We know it can have an impact. Basically also the test rate will be different, because if you take really the time and the effort to do a simulation study, you will stick a little bit more to your actual application. pH will be different, of course, and temperature will be different. You can avoid to generate the data at the 40 degrees C, because you don't need to cover 85% of all possible applications. And contact time also. Alicia, we thought of you. What is interesting there is that when you look at the data, and here basically you see all data and you can trace back the compounds. Here it's a simplified table, because based on the definition of the analytical evaluation threshold, everything under a certain level has been removed. We kept only the compounds that were needed to perform the tox assessment, and basically where it comes from, either the tubing, the filter, aseptic connector, or fittings and other adaptators. So if you summarize that, you see basically the compounds, where it comes from, what you can expect to go out of it when you are using water, when you are using the acidic solvent, and to compare, when you perform a simulation study, the picture is very, very different. By the way, it adds some compounds at very low level that can be expected, because it's always, and the standard extraction solvent are following the 80% rule. Basically, you expect to get as much as possible, but don't expect to get each and everything that can potentially go out of the component of your system. And basically, the levels also are very different. I circled the ones that where the difference is the highest. That doesn't mean that it was needed, because even with the levels before, with standard solvents, there was no toxicological risk. Why this study was done was more about, for some components, the data set was not available. So current, and I put questions, it can be challenges. Basically, of course, the questions that are all around on the market are basically what are the expectations of the authorities? Will they be happy with 665 and Bioforum extractables datasets, and or but all the feedback we get currently from the market are that really 665 is the relevant reference, which is very well understandable because it's USP. Now, some questions. There are some slight differences, especially about one extraction solvent. And the discussion becomes more open about, can you leverage the data from the 0.5 molar NaOH solvent from bioform, or do you need to generate the additional 10 pH 10? I don't have the answer for the moment. If there are some people that have, okay, we can always say I want the best, but again, we are discussing about, like you were saying, setting the bar at the right level and not over testing. So sometimes also you can have, for some product family, small differences in material of construction. I heard that from another filter manufacturer. There, basically, in my company, we had a specific filter format where there was a specific different PP material, and there, basically, it was totally retested. But only this part considered as a subcomponent, and just to check and to close a gap. What approach to apply with small parts with low risk? I heard that, okay, 665 low risk is there for them. We see different approaches, people tending to generate sometimes more data, maybe also having some semi-family information, sometimes some pooling approach, pooling a lot of different components and looking if there is a peak of concern emerging or if everything can be put aside. So this is not fully clear, and this can be some questions there. There can be some questions. Just taking, it's a little bit like we were discussing, sorry, I forgot to mention that, about the clearance of extractables. And Alicia, you mentioned the study that was published where basically a TFF is not clearing all extractables or all leachables. And that's raised the question, we need certainly to publish a lot more and then to come to a better understanding where we could have problems and where we could definitely, based on science, consider that these compounds can be neglected. But be careful, don't just consider because you have a TFF step that you can remove all the history of the process. It could work for the moment. It could work less easily in the future. Other question, and again, happy to hear about a COVID story where in COVID situation you can expect some more relaxed approach from authorities. Can there be some basically simplified approach for double sourcing or some comparative approach that could really allow us to do the right test, but not too much? These are questions that are currently will probably be answered in the coming years. And that's my first, I don't usually speak about sustainability, but of course, in the future, we will speak about sustainable material, meaning some part of recyclability somewhere in the manufacturing process of the parts, either at the level of the resin or in using some recycle portion when manufacturing the stuff. All that will need to be incorporated then in extractable studies. So, very simply, as conclusion, my message to you is that for the dataset to be considered historically, basically BPOG standardized protocol paved the way, but not really, now really USP 665 is the regulatory reference. Happy to, you see, Etienne, we are aligned in our messages. So that means that it's not, we are not at the end of the journey. There are a lot of data that still need to be generated to be aligned with 665, either medium or high risk. Risk assessment, similarly, we were used to use the BPOG best practices. Now we are more aligned to USP 1665, and that's the normal evolution, let's say, of the history. Be able to leverage suppliers' extractables data is of tremendous importance, but requires to be careful. Basically, we are committed for our own product in Cytiva to generate at least USP 665 data medium risk for all of our products. Regulatory requirements are evolving, and we will see what I have not personally read yet 383, because even if I started my first extractable study a long time ago, it's not anymore my main focus area of expertise. So, and definitely don't forget that even if you have issues with a paper exercise and current dataset, that doesn't mean that you have extinguished all options and you may want to go one step further. That might be sometimes the smartest solution. So I wanted to really thank my two colleagues that basically are one level above me in that field, who are based in the US, James Hadcock, my direct colleague, and Cherry Shi, who's working in his team. Thank you.